Assalamualaikum warahmatullahi wabarakatuh. For introduction, blood in fish plays a crucial role in transporting oxygen, nutrients, and waste products from the digestive system to the body. It binds to oxygen in the gills and releases it when transferring carbon dioxide from tissue to the lungs. Blood also helps in the distributing it throughout the body, regulating body temperature and transporting hormones for physiological process. It also helps maintain pH balance in body fluids, preventing harmful changes to bodily functions. Different fish species have unique variations in blood structure and function. Fish haematology research is crucial for understanding aquatic organisms' physical and ecological status. Eels offer unique opportunities for in-depth investigation due to their unique traits, geographical preference, and migratory behaviors. Studying eel haematology can improve conservation initiative, fishery management, and aquaculture technique, leading to better knowledge in aquatic species management. Objective: to determine the plasma osmolarity and blood components of Asian swarm eels, Monopterus albus, in response to body fluid balance. Second, to investigate how hematological parameter fluctuation correlate with eel physiological health, with the goal identifying typical values and potential markers for fragile and disease. Lastly, to analyze potential links between hematological measures and external variables such as water quantity and quality, temperatures, and habitat characteristics to get understanding into how eel reach to their environments.
Okay, so the materials used in the lab 5, which are Dorper, scissors, glass light, Asian swarm eel, scalpel, compound microscope to observe the red blood cell and white blood cell, syringe, appendix tube, vacuum microcentrifuge, and also the osmometer. For the first process is the blood sampling, where the samples of Asian swarm eels were provided, and then the fish were immersed in an anesthetic solution for 3 to 5 minutes. The fish were net to the port of fish holder once the fish were no longer responded to external stimuli. The outer mucus were removed using paper towels, and then the posterior end of the eel were cut with a scissor at 1/4 of the eel, and then the fish were massaged from the abdomen to the tail and collected the peripheral blood into a hair-perenized vacuum tube, as the fish were held tightly by its head vertically and immediately, and then slowly inverted the blood in the vacuum 2 to 3 times gently to mix the blood with the anticoagulant, and then the vacuum tube were stored on ice. Okay, the next process is the observation of blood layers, where the eel peripheral blood samples were liquid 1.5 ml from the hyperonized vacuum tube into an appendix tube and were labeled. The blood samples were spun in an appendix tube using microcentrifuge, and then it took out the tube carefully, observed and photographed the 3 separate layers, and then we labeled the layers accordingly. Okay, so for the third process is the determination of plasma osmolarity. So we collected the upper layer of fluid carefully, and then its osmolarity were determined by using an osmometer.
Pada bahagian result, terdapat dua sampel yang telah diambil di dalam makmal, iaitu sampel A dan juga sampel B. Sampel A adalah red blood cell ataupun RBC dan menunjukkan bacaan osmolality sebanyak 284 milliosmoles per kilogram. Manakala sampel B pula adalah white blood cell ataupun WBC dan menunjukkan bacaan osmolality pada 299 milliosmoles per kilogram.
Sel darah merah ikan ataupun RBC ialah sel bernukleus yang secara aktif menyumbang kepada homeostasis sistem imun dengan mengawal selia ekspresi beberapa set gen sebagai tindak balas kepada rangsangan. Mengandungi organel dalam sitoplasmanya mampu bertindak balas terhadap patogen yang menyerang dan bertanggungjawab untuk pertukaran gas. Sel darah putih ataupun WBC adalah sebahagian daripada sistem imun dalam ikan seperti dalam spesies lain. Sel darah putih ikan kebanyakannya terdiri daripada neutrofil, eosinofil, basofil, monosit dan limfosit. Sel-sel ini penting untuk tindak balas imun semula jadi dan diperolehi yang membantu dalam pertahanan terhadap jangkitan dan pemeliharaan kesihatan ikan.
Based on the result obtained, the osmolality sample blood B value is higher than sample blood A because based on observation sample blood B more clear is color in the sample blood A after insertion into the microcentrifuge, while the blood sample seems to be mixed. This is affected by the concentration of the blood because it becomes a diagnostic tool for assessing the health of fish population, abnormalities, parasite, and site of infection each themselves onto the canvas, providing a comprehensive health assessment. Hematological parameters emerge as vital indicator, allowing us to gauge the well-being of the aquatic ecosystem.
In addition, under the lens, the cellular landscape comes alive. Red blood cells, with their distinctive bioconcave shapes, become protagonists in the story of oxygen transport. White blood cells, the unsung heroes of immune defense, showcase diversity in form and function. Platelets, often overlooked, contribute their part to the symphony of fish hematological composition. Other than that, the oxygen carrying capacity of red blood cells takes center stage, unveiling its role in the respiratory efficiency of fish. The intricate dance between oxygen transport and environmental conditions becomes a revelation, underscoring the dynamic interplay between hematological adaptations and the aquatic needs.
To summarize, fish hematology is critical to understanding the physiological health of fish species and contributes greatly to fisheries and aquaculture management. Hematological indicators such as blood cell counts, hemoglobin concentration, and hematocrit levels that provide vital information of fish populations, overall health, stress responses, and disease status. Monitoring these metrics gives early warning signs of environmental stress, dietary abnormalities, or pathogen presence because fish health has a direct impact on growth, reproduction, and population dynamics. A true understanding of fish hematology is critical for sustainable fisheries management and aquaculture methods.